Hello students, welcome to this particular module. In this module, we will be discussing about the morphology, anatomy and reproduction of pteris. In morphology, we will be talking about its external features. In anatomy, we will be discussing the internal details, and in reproduction, we will be talking about the sporophyte and gametophyte generation. Before going into details, we should know the exact systemic position of this particular genus. It belongs to the phylum Pteridophyta, class Pteridopsida, order Felicalis, and family Densitaceae. So, this particular Pteridopsida has some salient features. What are these features? Firstly, the sporophytes are usually perennial in nature and it is differentiated into root, stem and leaves. The leaves are usually large and pinnately compound, and this is described sometimes as frond. Rachis are covered with brown hair called ramenta, and young leaves shows circinate venation. Sporangia are borne on the abaxial surface of the leaves or on the margin or tips of the pinnule. Now we will be discussing about pteris. Pteris has a cosmopolitan distribution, that is, it is widely distributed around the world, but it prefers to grow in tropical and subtropical areas. Most of the species are terrestrial in habitat and they grow in well-drained soils. The name pteris has been derived from the word pteron, which means feather, due to the feathery appearance of the leaves. These leaves, known as megaphylls, are known as fronds. The thin or thick-walled sporangia are borne terminally in the axis of the fronds or below the adaxial sides of the frond. This is the sporophytic plant body, and the sporophytic stage is the dominant stage in the life cycle of pteris. Now this sporophytic plant body is differentiated into roots, into rhizomatous stem and the leaves, which are known as fronds. Coming to the root, the primary root that are present are short-lived and is soon replaced by the adventitious roots. The roots are slender, black and arise from ventral side of the rhizome or are present all over the surface of the rhizome. Now, if we look internally into the anatomical details, we will see that the root is differentiated into an epidermis, a cortex and stele. The outer layer is the epidermis, which consists of a single layer and are formed of thin-walled cells. Some cells form the root hairs and are present in the epidermis. Just below the epidermis lies the cortex. This cortex is multi-layered and is differentiated into an outer cortex and an inner cortex. This outer cortex is formed of parenchymatous cell, whereas the inner cortex are sclerenchymatous. There is a presence of an endodermis, and the endodermis has the Casparian thickenings. The stele is present in the center and it consists of xylem and phloem. The xylem is either exarch or diarch and is surrounded by phloem on either side. Outside the stele is a thin layer of pericycle. This is the picture in which we can see that in the central region the stele is present and here is the cortex and the outer layer is the epidermis. Now coming into the rhizome. While I was showing you the sporophytic plant, you have seen that the rhizome can be creeping or can be erect. The rhizome can have branches or can be devoid of branches. This rhizome is differentiated into nodes and internodes, but whole of the surface is covered with scale. The growing point of the rhizome is covered with fine hairs and these hairs is known as ramenta. Coming to its internal details, we will see that if we cut a TS of this rhizome, we will find certain structural details. In the outside layer, there will be a cuticle. This cuticle protects the plant from desiccation. Just below the cuticle is this epidermal layer. This epidermal layer that is present is single layered. Just below the epidermis, you will find this hypodermal layer or the hypodermis. This hypodermis is formed of sclerenchymatous cell, and these, as you can see, is a few cell thick. Just below the hypodermis lies the cortex. In this structure, you can see that cortex forms the major portion of the internal structure of rhizome. The cortex is formed of parenchymatous cell. Coming to the stele, which is present in the middle portion, there is a certain peculiarity about the stele of pteris. The stele can be of many types. That is, the stele is diversified in this particular genus, starting from solenostele, dictyostele, sometimes it may be protostele also. But usually, meristeles are present. The meristeles are present in two rings. One is present in the outside, and in the middle one, we find the meristeles are little larger and are usually 2 or 3 in numbers. But the outer ring formed of small meristeles are usually large in number. Each stele is surrounded by its own endodermis. Now we come to the leaves. The leaves are borne on the upper surface of the rhizome. Now, during the formation of the leaf, that is, when the plant is in young stage, the leaves are spirally coiled and they show sarcinate venation, that is, the very typical venation of true ferns. The leaves can be unipinnately compound or multipinnately compound. Sometimes it may be also decompound, but usually a long rashes is present. The pinnae are small near the base as well as towards the apex. The leaves are usually fertile and they contain the sori along the ventral side except in the apical region. The rashes is traversed by a C, a U or a V-shaped leaf trace. The lamina is bifacial. The mesophyll cells that are present may not be differentiated. A concentric vascular bundle having a distinct bundle sheath is present in the midrib. Here is the structure of a pinnae, and you can see that the sorus are arranged along its margin. Now coming to its reproduction. This pteris reproduces with the help of spores, that is, it completes its sporophytic generation, and after the completion of the sporophytic generation, the gametes are produced and the gametophytic generation start. The spores produced in pteris are all alike, that is, the pteris is a homosporous plant and the sorus is known as Soanosaurus. The Soanosaurus of pteris are marginal and are borne continuously on the vascular commissure connected with the vein ends. Thus, the sporangia of pteris form a continuous linear sorus along the margin, and these Soanosori are protected by a reflexed margin, and this margin is known as the false indusium, which is a very characteristic feature of this particular genus. The sori are of mixed type and are intermingled with many sterile hairs present in between the pinnae. Now, this is the structure where we see a pinnule showing the sorus. Here you will see 3 different stages of sorus are shown. This particular sorus or sporangium has been dehiscent. This is a mature sporangia and is on the verge of dehiscence, and these two present at the both of this side are the young sporangia that is present. This is the fall inducium that protects the sorus, and this is a hairy outgrowth that is present. Now we will be discussing about the developmental stages of sporangia. The development of sporangia of Pteris is leptosporangia type. That is, it is formed from a single initial. Let us first consider this word diagram. We will see that the sporangial initial is first divided into the upper cell and the lower cell. This sporangial initial can be any of the superficial cell. Now the sporangial cell is divided into upper cell and the lower cell. The lower cell do not take part in the formation of sporangium and it forms the stalk. The upper cell divides to form the apical cell. Further division of the apical cell takes place. This forms the jacket initial and the archisporial cell. This jacket initial further forms the jacket, and archisporial cell is divided into the tapetal initial and the primary sporogenous cell. Tapetal initial will be forming the tapetum layer that function only as a nutritive layer, and the primary sporogenous cell further divides to form the spore mother cell. Now let us see this particular picture. See, this is this particular superficial cell that is functioning as the sporogenial initial. The first division forms the upper cell and the lower cell, and this is an unequal division in which the upper cell is slightly larger than the lower cell. Further division takes place, but the lower cell remains intact. Slowly the apical cell is formed, and from the apical cell, the jacket initial and archisporial cell are formed. This jacket initial forms the jacket that is protective in function. It has the tapetal layer, and inside is the sporogenous cell which further form the spore mother cell. This is the stalk that the lower cell forms. These spore mother cells further form the spores. Now coming to the mature sporangium. This mature sporangium has a long stalk, and this long stalk further terminates into the capsule. The jacket of the capsule has three distinct layers. It has an obliquely vertical annulus which incompletely overreaches the sporangium. So there is this annulus which is oblique vertically placed. Then we come to the stomium. The stomium is a thin-walled and is radially arranged. Third are the large parenchymatous cell with undulated walls. Here we can see that this particular sporangium has daist, and these are the spores. As we have discussed before, these are homosporous. So all the spores that are produced are alike. These spores are triangular in shape and it has a distinct triradiate mark. Now we will be talking about the gametophytic generation. But before that, this spore should germinate. So by absorbing water, the spore swells up. The spore has two distinct layers. One is the exine and the inner one known as the intine. The exine is very much sculptured. Now this wall breaks, that is the exine breaks, and the inner contents comes out in the form of a germ tube. Now further division takes place. The germ tube forms the first rhizoid and the first prothallial cell. The apical cell further divides and a prothallus is formed, which is a spatulate-like structure. Thus, with further division, a mature prothallus is formed. The prothallus is corded, dorsiventrally flattened, and is photosynthetic. The prothallus is formed of parenchymatous cells, but there is a difference in the density of the parenchymatous cells. The parenchymatous cells are not evenly distributed. The cells near the margin are single layered, but as we move towards the center, it becomes more dense. The rhizoids are formed from the ventral surface. This is the structure of a mature prothallus. This prothallus is monoecious and protandous. By monoecious we mean that the male and female gametophyte are formed on the same plant. You can see here that the female gametophyte, that Archegonia, are present near the apical notch, and the antheridia, which are larger in numbers, are present in the basal region or the lateral region, and they remain intermingled with the rhizoids. Now, there will be the development of this male gametophyte and the female gametophyte. So, the development of the antheridium. So, how this antheridium will develop? This antheridium will develop from the superficial cell present on the ventral surface. So, it will act as the antheridial initial. This antheridial initial will again be divided into an upper cell and a lower cell. There will be an absorption of water, and that will increase the turgor pressure. Thus, the upper cell will become a dome-shaped. This dome-shaped cell will form the dome cell and the primary androgonial cell. Further division of the dome cell will take place, forming the cover cell and the second ring cell. The lower cell that was formed before will form the first ring cell. So, this is the second ring cell, and the lower cell formed the first ring cell. Now, the cover cell and the second ring cell together will form the jacket. Primary androgonial cell will form the androcytes, and these androcytes will be metamorphosed into antherozoids. Coming to this picture, this is just the same which I have explained, that this antheridial initial will be there. There will be this unequal division forming the first ring cell and the upper cell. Due to the increase in the turgor pressure, a dome-shaped cell will be formed. The further division of this dome cell will give the dome cell and the androgonial cell. It will form the cover cell and the second ring cell, and slowly this division will form. The outer jacket layer is formed, the androcytes are formed, and slowly the flagellated antherozoids will be formed that would be released by rupturing of the wall. Now coming to the development of the archegonium or the female gametophyte. This development takes place again from the superficial cell that acts as the archegonial cell, forming the inner cell. Here the outer cell will form the primary cover cell. First primary cover cell will be formed, that will form the neck initials. The neck initials will be forming the neck cells. The inner cell will again be divided into two parts. One will form the basal cell and the other will form the central cell. The central cell will further form the primary canal cell and the primary venter cell. This primary canal cell will form the neck canal cell, and the venter canal cell will form ventral canal cell and the egg cell. So, here it is the diagram that from this archegonial initial, how this mature archegonium is formed, in which there is this neck portion, there is this venter, and at the bottom there is the egg. Now, the fertilization will take place when both the antheridia and archegonia have been fully developed, proper egg and antherozoids are being formed. Fertilization in Pteris takes place in the presence of water. So, there must be water present between the lower surface of the prothallus and the soil. The antheridium will absorb the water, it will swell, and the cover cell will break, releasing the antherozoids in the water. Now the antherozoids will swim towards its female gamete. How? There is a particular attractant that this female gametophyte secrete. This is the malic acid. So, malic acid of the archegonial secretion will help in attracting the male gametes by chemotaxis. The male gamete will enter the archegonial through the neck cells and will ultimately fertilize a single egg. The type of fertilization in Pteris is oogamous type, and once the egg is being fertilized, the archegonial venter will form a cap like calyptra around the fertilized egg to protect it. Now after it has been fertilized and the zygote is being formed, it will form a new sporophyte. So a new sporophytic generation will come up. The first division of the zygote is vertical. So a two-celled structure will be formed. The second division is transverse. So after the first division, the second division is transverse. So a four-celled structure is formed, or now the zygote has attained a four-cell stage. Further division of this zygote will take place, and ultimately it will result in an embryo consisting of 32 cells. This embryogeny is endoscopic and no suspensor is formed. The hypobasal cell of the embryo will form the stem, the apex and the foot. That means now it will be divided into the hypobasal cell and the epibasal cell. So, the epibasal half will form the cotyledon and the root. It is being noticed that in the young embryo, the root and cotyledon grow more rapidly than the shoot. This is the picture where we see the development of the embryo. Starting from the two-cell stage, this has gone to this four-cell stage forming the epibasal hypobasal cell, and ultimately this cellular structure is being formed having the foot, root, cotyledon, stem, and even the prothallus are also being formed. So all this forms the life cycle pattern of Pteris. So what do we mean by life cycle pattern? That is, it has a sporophytic generation, that is 2N generation, and a gametophytic generation, that is the N generation, and both these phases alternate in a life cycle of this particular genus. So if we start with the sporophytic plant, as we have studied, that this sporophytic plant has the sporophyll, and here present are the sorus. If we get a TS of this pinnae, we will see that the sporangium are present. Pteris is a isosporous, that is the spores produced are or similar. So the spores produced are the spore mother cell, and from the spore mother cell the spore is produced. Once the spore is produced, by the process of meiosis it enters the N generation. From the spore, the gametophytic plant body or the gametophytic thallus is being produced. This gametophytic thallus consists of the female gametophyte present at the top and the male present below. So the male will be forming the anthrozoids and the female will be forming the egg that is present at the lowermost region. Once water is being present and the attractant is being released by the archegonium, this anthrozoid will come and will fertilize this egg. This fertilized egg will form the zygote. Since both this sperm and this egg are N, so it will form the zygote, which is 2N, and this zygote will further develop into a young sporophytic plant, as we have studied before, that this young sporophytic plant will slowly mature and will form this sporophytic plant body, thus completing the whole life cycle of Pteris. So in this module, what have we studied? We have studied the morphological features or the external features of this particular genus, starting from the root, the stem and the leaves. The stem is rhizomatous and the rhizome can be erect or maybe creeping. After that, we talked about the anatomical details. The anatomical details of the root, the anatomical details of the rhizomatous stem. We have seen what type of stelae are present in the root and in the stem. After that, we came up to the leaves. We have seen that the leaves showed circinate venation, that is, it is a true fern character. Then we talked about the sporophytic plant body, how the spores from the sporangia are being liberated and how they germinate to form a gametophyte. That gametophyte was a dorsiventrally flattened structure bearing the antheridia and the archegonia. Then we talked about the development of the antheridia and development of the archegonia. They got developed, they produced the androcytes or the sperms and the archegonium or the egg. So both the sperm and egg got fertilized when there was water and they formed the zygote. Further division of the zygote formed the new sporophyte, and that sporophyte plant developed to a complete sporophytic plant body, thus completing the life cycle of Pteris.